Perfect. Okay, so this is the DNA replication part of the lecture. So last time we talked about structure and what DNA is made of and how it looks. We talked about this nice model here. Here's our double helix of our DNA. Today we're going to go through all of the process of how we replicate this. When we are replicating it, we need to replicate it so that when the cell divides in mitosis, we have enough DNA that both of those two daughter cells get exactly the same amount. All of the DNA is copied. All of it is there. Those cells can be successful. So this is happening during interphase. And interphase is 3 parts, our G1, S, and G2. And it happens during that S phase. There might be some blanks on there. So DNA has to be copied before a cell divides. It's during the S phase. And those two cells that come out of mitosis need to have the identical amount of DNA. We're going to do a couple of slides and then watch a video, and that's when that will start. Yep. Sorry, skipped too many. There we go. All right, so here we had our cell cycle that we were talking about during the mitosis lecture. And we have interphase here. And it's happening during the second S phase. Remember, our G1 phase was when the cell was growing pretty rapidly. The G2 phase, we're replicating all of our DNA. And then, the S phase, we're replicating the DNA. And then G2 phase, we're growing a little bit more. And we're making sure all of those other organelles and the proteins and everything that we need for mitosis is all prepped and ready. Okay. This is happening in the nucleus of eukaryotes. Remember, prokaryotes don't have a nucleus. Eukaryotes do.
Wait, where does it happen in the prokaryotes?
So the prokaryotes, we have a lot of our DNA in the prokaryotes just kind of floating around inside the cell. There isn't like one contained area like a nucleus.
So, but it still happens?
It still happens. It's still going to replicate because the cell still needs to divide. There's just no nucleus for it to happen. So it's just happening kind of in the cytoplasm. Okay. All right, so we're going to watch this video here. Stop.
Did they write anything down?
Nope, we're just going to watch it through first. And then we'll keep kind of going back to this video and we'll start and stop it along the way and kind of go through the different parts of it. Okay? No, it's not. We'll just do the volume down here.
A pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase 3 can then add deoxyribonucleotides to synthesize the new...
Okay.
Let me just do something quick here. You gotta go to systems preferences.
Oh, okay.
Should be a tag.
You have to save it in?
Should work. This is going to be super loud.
Okay. Oh, the volume's off right now.
They are anti-parallel. They are oriented in opposite direction.
All right.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Wonderful. It's a lot. Yes.
Wait, so is that all makes sense?
Yes. So this is the entire process of replication. It's a lot.
One that just like kind of goes down the line, but then the other one has like 8,000 steps.
Yes. So we have the top strand that we call the leading strand. That's the nice strand that just kind of flows. The bottom strand is called the lagging strand, and that's a pain in the butt to deal with because there's so much going on. Okay. It's a lot. We're going to break it down and take it kind of step by step here, and then we'll put it together.
And then another synthesizer, and then this number 3, whatever. And then another thing takes off the synthesizer, and then another thing puts in the DNA.
Yes. All right. So let's start at the beginning then. We have what we call this origin of replication. This is where this replication is going to start. It starts to make a replication fork, which is this Y shape in the DNA. Those new strands are going to start to replicate at that fork. Yes, replication fork. So the origin of replication. Do you guys need another second, or can I go to the next slide? Sure.
Where is the origins of replication?
So the origin of replication, if we're looking at our DNA here, if we untwist it, it starts to look like this ladder, right?
Yeah.
So then we're going to unzip in the middle, and it starts to kind of look like this. So we create kind of this bubble. Here's our ladder part, and we're literally just breaking it apart in between the nucleotides and unzipping it. this is where our replication fork is.
Oh,
So we actually have a bunch of these replication forks that are happening in your cells right now, all the time. And in eukaryotes, we have a bunch of what we call these replication bubbles. So this process of unzipping the DNA and getting it ready for replication doesn't just happen in one spot. It's happening all over the place in your DNA strand. And it makes like these little bubbles where replication is happening. In prokaryotes, there's only one replication bubble. In eukaryotes, we can have a whole lot of replication bubbles happening all at once.
So in prokaryotes, like in bacteria, they only have one thing that's being replicated at a time.
Yeah, we're only unzipping one part of the
In every
the
cell,
DNA.
or only at one cell.
In every cell.
Okay, so like only one piece of DNA can be replicated in a cell at a time.
Yeah, only one part. So here we have our DNA strand, and it's kind of split open and having bubbles along the way at different parts. This is the eukaryotic cell. In a prokaryote, we'd have that ladder, and it would only have one bubble in the middle of it, and that's where the replication would be happening at that time. Yes. Yes.
Okay, and then in the eukaryotes, you can have multiple. So like there's like one strand of DNA, and it's like that one, and it can like open and then close and open and then close.
Yep.
Wait, we're filling out this whole package by today or on Friday? Or today and Friday?
Hopefully we're going to get through all of it today. If we need to do a little bit on Friday or a review on Friday, we can. Okay? So the thing that's doing the unzipping and making these replication forks and this origin of replication is this enzyme called helicase. Helicase. So helicase in my little DNA model over here is going to be an enzyme that forms right here. You can think of this as like the zipper pull for your DNA. It's actually going in and it's breaking the bonds between the A's and T's and the G's and C's. It's splitting it right down the middle, and it's unzipping that whole strand of DNA. After we unzip the strand of DNA, we have to hold it open so that way it doesn't stick right back together. These single-stranded binding proteins, or we call them SSBs, they're sometimes referred to as that, are like these little proteins that stick on the outside edge here. And their job is to just hold these two strands open so that they don't stick back together. If they stick back together right behind helicase, we won't have a big enough replication bubble in order to start doing any of the replication. We won't be able to copy it because there's no room. We need to have these hold that DNA open so that way we can replicate. Okay. The next enzyme that we're going to be talking about is called topoisomerase. Topoisomerase is a protein or an enzyme that is way out in front here. And topoisomerase's job is to kind of unravel this DNA. If we have it all coiled up like this and we're trying to unzip it, it's really hard to be able to unzip it evenly and nicely. Topoisomerase is going to come in and it's going to untwist all of it, so that way it's a lot easier for helicase to unzip it. It also reduces a lot of the strain and tension. If we don't untwist it first, we can have a lot of supercoils on the side, and that's where a lot of those breaks can happen. What was that? Reduces strain and tension on the DNA.
So it's just like untwist it.
Yeah. Topoisomerase goes out in front and untwists it. Got it. Good. You need a second. One of both. Okay, let's go back to that video and we'll jump to this part then. Okay. We'll zoom back here to start.
It begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it.
Okay. Good so far?
Yeah.
That's the first part.
What about the topoisomerase?
Topoisomerase, they don't have it in this animation, but it'd be sitting right out here, right in front of the helicase. In order, I don't think my thing isn't working anymore.
DNA polymerase 3.
So it would sit right out in front of the helicase and untwist it so that it doesn't get too much tension and strain on that DNA.
Okay.
Cool?
Got it.
Wonderful. All right, so that's our first part of it. Is getting it ready to replicate by unzipping it and untwisting it. The next part of it is we're going to lay down what we call a primer. So we can't just start adding DNA nucleotides. We won't be able to start with that. We have to start with what's called a little primer. So an enzyme called RNA primase comes in and it lays down like 10 base pairs of RNA. Just a little chunk of RNA in there. From there, then we can start adding on some of our DNA nucleotides. So this DNA polymerase is then going to start adding on nucleotides. And it's going to start forming down the line.
So it adds like, it tricks it into being like, oh, this is what you need. It's like photocopying.
It is kind of like photocopying. So what we're doing is we're going to try and replicate each side in order to make 2 copies of our DNA. Okay? We got to put down this little chunk here first. That's our RNA. And then once we have that little RNA primer, we can start adding on some of our DNA nucleotides. The DNA polymerase that's adding on those nucleotides can only add them in a 5' to 3' direction. Put a big box around this or a star or highlight it. This is an important part here.
Which one?
That it's built in a 5' to 3' direction.
Whoa, how do we get there so fast?
Because the last slide was a picture of the full thing again. It was a picture of this.
This one.
This one. That one? There you go. So we had our RNA primer, puts down the little chunk of RNA, and then our DNA polymerase is going to start adding DNA nucleotides. Juan, do you need another second? So we're going to add those nucleotides to the 3' end of our DNA. Remember when we were talking about numbering our nucleotides here, and we were numbering our carbons 1, 2, 3, 4, and 5? Does that look familiar?
Yeah.
Okay. The only way that we can add nucleotides is if we bring another one in and link it into the bottom here. So we're going to have our next nucleotide come in. And it's got a link to that 3rd carbon there. It's the only way that we can build them on. So what this is showing then is that we're starting at a 5' side. This side is the top here. This is our 5' end. And we're going to build in the direction of the 3'. So we're going to keep adding on to the 3' side. That's how we're getting this whole, it has to be built in a 5' to 3' direction. Did I lose you guys? You still with me? Okay. Huh? So this is, I'm numbering the carbons here. Here? Yes. So our original strand, this side was 3' to 5'. When we're laying down new nucleotides, remember our complementary strand is a 5 for this one, and we're laying it down in the 3 direction, 3' direction. Here? So the template is 3' to 5', but when we're building it from 5' to 3'.
I see what you're saying.
I get it. Yeah, I get it.
So you read the template strand?
Yeah.
So that strand is on top. You read it 3 to 5, but you build the new one 5 to 3 because that 3' carbon is where the new phosphate has to attach to. So if you were to take, so basically what she's trying to show you is that if you take this right here and then turn it like this, does that make sense? Right? So you just tilt it this way. So your 5' is here. Right. So remember, DNA has to be anti-parallel. So the new strand that you're building has to be anti-parallel to the one that's already your original template strand. So on this one down here, if this is 5', your new strand will be 3 and 5 will be here, but you can still only build new nucleotides 5 to 3.
And that's why you have to do it in small chunks.
Correct. So that's why you have the leading strand at the top is always, well, it's not at the top, I should say that. The leading strand always works towards helicase. And the lagging strand will always work opposite of that because it can only build still in this direction. And it only will work as fast as this opens up.
Where's the 5?
Where's the what? So the 5 is just talking about the number of carbons, and this is why that is so important when we look at the nucleotide sequence, is that your first carbon, this is actually how we just number the carbons. 1 is always attached to the base. 3 will always be attached to the next nucleotide below its phosphate, and 5 is always attached to the phosphate. So when we talk about a 5' and a 3' end, we're really talking about the 5th carbon and the 3rd carbon. So if you see the number here, right, 5', all it's saying is that this carbon is now lying in that direction. Does that make sense? So like I said, if you actually just took this picture right here and go like this, the 5 now goes at the top, and the 3 will be at the end. Now again, that's important because when we add new nucleotides, we can only build it this way, from 5 to 3. So going this way is we're going to keep adding new ones on. Does that help?
Yeah.
I understand what you were...
I was just confused with the labels actually meant. It means like which carbon is.
Right.
Yes.
And one strand isn't the 5 strand. And one is
No.
Correct.
Right. It's the direction that is being read and or built in.
Yep. Yeah, so we have those 2 strands that are anti-parallel, so they're both looking like this. It's just talking about which direction it's pointing. Okay? Did you have a question before you go?
No, I'm good.
Okay.
And then
Wonderful.
they will be on campus, right?
Yes. Okay? So we're constantly building in this 5' to 3' direction. Make sure you put a star around that or a box. That's an important part. Okay? So here was what we were talking about with numbering our carbons. So we have our carbon 1, 2, 3, 4, and 5. And we're always going to be adding our carbons, our next nucleotide, to the 3rd carbon. So if you look at this strand then, it looks upside down. It's the inverse of it. Our 5 side is actually down on this one. So we're going to constantly be adding to the top for this side of it. So we always have to add to the 3' side. Okay. All right, so let's look back at that video.
DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments.
Okay. So we'll go talk about the leading strand and the lagging strand a little bit first, and then we'll come back to the video and watch those in action. Okay? So this leading strand, in our case on the board here, this leading strand is the top strand. The leading strand is going to work 5' to 3', and it flows in the direction of helicase. So as it's unzipping, it can just keep working and keep adding those nucleotides. It doesn't have to start or stop.
What are the little lines?
Which little lines? These little ones?
Yes.
Those are like our A's, T's, G's, and C's. Those are our nitrogenous bases.
Nice.
So what it does is we normally look at our DNA as kind of like this ladder here,
And then
and we just
split open.
unzipped it right in the middle.
Sick.
Okay? So if you want to write down in the notes that your leading strand is going to follow the helicase or follow the replication fork. So it's going to be flowing in that direction, which is why the leading strand is the nice strand, because it just keeps going. We don't really have to worry about that one a whole lot. Alright. The other side then is what we call the lagging strand. This one is kind of the pain in the butt sometimes. So that's the one in this case on the bottom. And it's kind of the pain in the butt because it has to work against the replication fork. So it's going to work going, in this case, to the left, away from the helicase. Okay. So because we can't flow with the helicase and with the replication fork, we kind of have to do it in these little short chunks, these little segments. So when we're building from 5 to 3, we have to first lay down, what's my DNA? We have to first lay down a little snippet, our RNA, and then we're going to start adding on DNA, but it can only go in this direction, because we're only going from 5' to 3'. Once it gets so far, then it has to stop, and it has to jump up, and go to the next part, and lay down another little primer, and then build the DNA until it gets to that first primer, and then it has to stop, and it has to disconnect and jump up. It's kind of a pain in the butt. These short little segments then are called Okazaki fragments. was named after two Japanese scientists. They were actually married, which is kind of nice because then it's just Okazaki fragments and not Okazaki hyphen something something fragments. Most of the time it's based off of whoever found it. Sometimes scientists are really nice and base it off of something that, a Greek or Latin name that means whatever it does. But a lot of times scientists just name it after themselves.
Selfish. They are selfish, yeah.
So we have these Okazaki fragments, and these are little chunks that we're building. So we can't just flow it all together, we have to do it in these little pieces. Once we get all of these little pieces, then we have to stick all of the pieces together. In order to do that, we need to use a couple of enzymes. The first enzyme, what color should I do? We'll do orange. We haven't done orange yet. The first enzyme that we're going to do is we're going to use what's called RNase H. And RNase H is going to attach and it's going to change those little RNA primers into DNA nucleotides. We can't just have RNA sitting in our DNA strand, we have to change it into a DNA nucleotide. So this is going to come along and change these 10 base pairs of RNA into our DNA. Yes, it will also come up here. Yes. Yeah, getting close. Okay. So that's our first enzyme. So we got this RNase H that's going to change all of those little RNA primers, all those RNA starters, into DNA. And then we're going to have, what color? We'll do this one. Then we're going to have another enzyme that's going to come in here, and it's going to glue all of those little Okazaki fragments together. That enzyme is called ligase. There we go. So the enzyme ligase is going to stick all of those Okazaki fragments together. So it's one continuous strand again. Remember, the top strand is still one continuous strand. It's been going along with helicase the entire time. It's all nice and neat and pretty. The bottom strand, we got to do a little bit more to make sure that those get changed over and that they all get glued together. Awesome. All right. Let's look at the video real quick before we go on to proofreading.
When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it.
This could also be called DNA polymerase I. We're going to use the name RNase H instead, just because that way it's not as confusing with the two DNA polymerases. Okay? In some videos, you'll see it called DNA polymerase I. We're going to use the term RNase H.
With DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
All right. So, are you guys good with the lagging strand and the leading strand? This is the next little confusing bit. So we were talking about we were talking about these two. These are our DNA polymerase. They're the ones that are laying down all of those new nucleotides, right? We're good with that? In a lot of depictions, it's going to show these as two separate parts. I'm shaking the video. That's fine. I just thought it was the system acting up or something. Okay. So a lot of times you'll see these as two separate units. What it's actually showing here is that those two units are attached and that the DNA is what's moving in between. So our DNA doesn't necessarily always stay in this nice flat view. Sometimes it can twist and go into the DNA polymerase that way. So that's just what this view is showing. Okay. You guys all right with this?
Sure.
You have any questions before we jump over to proofreading? So then looking at this, proofreading. Doing this process of replication is usually pretty smooth, but we can have a lot of errors that can happen. Either a nucleotide can come in and it doesn't match up, so an A and a G pairs together by accident. Or we can miss a nucleotide and it'll just be a blank. Any of those errors can mean that there might be proteins that aren't formed correctly. It can even mean that there's some mutations that are happening in our DNA. So we want to make sure that we catch those errors as soon as possible. We have about 1 in every 10,000 base pairs that makes an error. From that, we have some of our enzymes and our proteins that are proofreading it and double checking everything. You want to pull up a chair? There's one over on this table here.
No, Maggie's fine.
Okay. And then we have these enzymes that are checking in on all of our mistakes, and they're going to fix all of our mistakes. And that brings it down to about 1 in 1 billion base pairs that makes an error. This error can mean that there's a mutation. It can mean that there's just something that's formed that's non-functional. It can mean that there's something as serious as like a cancer, that this becomes a cancer cell, although that's very, very, very rare.
On a scale of 1 to 10, how rare is it?
Well, think about how often this is happening in your body right now. Your cells are constantly replicating and constantly dividing, which means that we're constantly trying to replicate our DNA. So it's happening all the time right now. How many of those times become a cancerous cell is very slim. Okay? So a scale of 1 to 10, by 10 being it happens all the time and 1 being it never happens.
Yeah.
Like a 1.1.
Nice. That's like 1 in 100 chance. That's not very rare.
Exactly. Well, no. 10 being it happens all the time. 1 being it never happens. Like a 1.1. Okay? I think this is one of the last kind of things that we need to talk about with our DNA replication. It's this idea of our DNA replicating in a semi-conservative manner. Okay? So semi-conservative was presented by Watson and Crick, who you remember were the ones that kind of figured out all of the structure of DNA. They came up with this idea that our complementary strands in our template are going to have 1 of each in our new cells. So when we split our cells, we're going to have 1 of the original DNA strands and 1 of the new DNA strands. This way, we can always go back and check to see if we made any errors because we have the original template. If we had both of the old strands in one cell and both of the new strands in another cell, we might make a lot more errors. Okay. Did you have a question? No? All right.
Holy cow.
Yeah. You could have cells that still have your original DNA strand from like one of those first cells.
Really?
You could.
That's
Yeah.
so cool.
Because we're always having one strand of the original and one strand of the new going into our $2 source. Okay. Next thing here is that we can still have a lot of errors that are happening just from us being out in the sun, us being in contact with radiation, us being in contact with chemicals. This is why you wear your sunscreen. Ultraviolet radiation, you can get that from the sun, from just being outside, walking in and out of the buildings, being outside playing sports, being outside at the beach or the pool. Wearing your sunscreen is going to help reduce the amount of radiation that your cells get, which then reduces the amount of errors that can happen in your cells.
Wait, so when you get like sunburn?
It's not necessarily sunburning that's bad for it, it's the radiation that causes it. So that's why they're always saying the damages of like skin cancer. Skin cancer is caused because there's so many errors that have been made in your DNA because of this radiation. Okay. Oh, sorry.
You have the question on the back, even though the answer is right there.
Yep. So we only have like what? One slide left? Two slides left?
One, two.
Two slides. All right. Yes, Kai. All right, guys, hold on. I can't hear Kaya. Hold on. Yeah. So this radiation could damage the strands. It could make there be a nucleotide that doesn't bond and kind of falls off. It could make those nucleotides that are replicating mismatch.
You're losing them. Not me though.
Okay. All right. Are enzymes that are perforating everything. Are repairing. All right, last slide. What is the complementary strand of this here?
5CGDAT.
Well, why does it have the answer on
Oh, I think I actually know this.
Yes.
I think I can know this.
Yep.
So the green side.
So if we switch it around, very good. All right.
Let's
We can
go,
put the
cats.
They might not go in right away. Yeah.
Okay, and so when
Just like this one. Just like I have.
Also, we know